Hello everyone and welcome back to Clinical Cousins YouTube channel. Today we're going to go over Audition, and we are not trying out for a play, we are seeing how we hear. So what are sounds? Well, it may sound like a simple question, but sounds are simply waves of pressures that have a certain loudness, which are measured in decibels, and a certain vibration frequency or frequency that is measured in hertz. So our ears transduce or convert these sound waves into electrical and chemical energy, which then travel to our brain where they are interpreted as sound. So I mentioned earlier that pressure is measured in decibels, which is abbreviated as dB. So this is how loud something is. Decibels are calculated by taking 20 times the log of the pressure being measured over the reference pressure. So any sound over 100 decibels can cause hearing loss. So this would be like if you're standing right by a subway and it's rushing by you, it's really, really loud. Or you're at a loud rock concert or rap concert. Any sound over 120 decibels can cause physical pain in your ear. This is like standing behind a jet engine while it is about to take off. You should know that the human voice is around 65 decibels. Sounds also have a specific frequency. We measure this in hertz or Hz. The hertz are increased as cycles, as speed of vibrations. So the higher the hertz value, the higher it sounds, and the lower the hertz value, the lower it sounds. We can hear sounds with hertz between 20 and 20,000. Conveniently, we speak at around 300 to 3,500 hertz. This would be very bad if we spoke, but then our ears couldn't hear it because it was out of range. But we know that this is not the case. So before we know how sound is heard, we have to know some basic anatomical structures. So the external ear, this is what we can see. It consists of an outer pinna and an external auditory meatus. This is a big, challenging phrase, but really it just means the outside part of the ear that has a hole in it. A meatus is a mouth. So we have our pinna and our external auditory meatus or mouth. This is our ear hole, it's our ear canal. And we should know that it's normally filled with air. So the pinna acts sort of like a funnel that funnels sounds into the ear canal, into our external auditory meatus. So next we're going to learn about the middle ear. This begins with the tympanic membrane. Remember that tympanic just means a drum. This is why it's often called the eardrum. The sound pressures are beating on our tympanic membrane. So the eardrum is connected to a series of bones called the malleus, the incus, and the stapes. In fact, the stapes is the smallest bone in your body. So these three bones connect to each other in such a way that the stapes eventually connects to a footplate in the cochlea. So the cochlea is part of the inner ear. We have the external ear, the middle ear, and now the inner ear. So we should know that the middle ear is normally air-filled, and that the connection between the stapes and the cochlea is called the oval window, which is shown here in blue. This brings us to the inner ear. The inner ear is made up of a bony and a membranous labyrinth. Remember that a labyrinth is sort of just like a web maze that you would see, maybe like a corn maze at Halloween. It has a lot of different canals and pathways running through it. It is important to know that the inner ear is normally filled with fluid. The cochlea is this spiral-shaped structure shown here, and it contains ducts and special sound receptor cells called the organ of Corti. So it is important to note that there are two different types of fluid that fill our inner ear. So first we have perilymph. Now perilymph is sort of similar to extracellular fluid, so think of sodium. Now endolymph is similar to intracellular fluid, so think of potassium. This will be important later. So how does auditory transduction work exactly? How do we take sounds and then convert them into energy, which is then passed along to our brain? Well, our cochlea contains specific receptor cells that are collectively called the organ of corti. So what I've done here, I've taken just a little section of the cochlea and I've drawn it out really big so we can see what's going on. So we have 3 scalas, or sections. So we have the scala vestibuli on top, the media in the middle, and the tympani on the bottom. All of the important transduction takes place in the media. So these hair cells, which are shown here in blue, these hair cells reside in what's called the basilar membrane. You can remember it, it's the base. Now this basilar membrane of the cochlea contains the cell bodies of these hair cells. These hair cells have little cilia, little itty bitty hairs that attach to a tectorial membrane. Tectorial means roof. So what happens is that sound waves, these sound pressure waves, enter the external auditory canal and they slam against our tympanic membrane, which vibrates the auditory ossicles or bones, and these auditory ossicles work to condense all that sound onto a tiny space which is called the oval window. This sound wave vibrates the fluid in the organ of corti, so in the scala media. So this is where we are. We've had some sound waves and it's vibrated everything up until this point. So because the basilar membrane is like gelatinous jello, it moves back and forth. These cilia are firmly attached to the hard tectorial roof membrane. When the body of a cell moves one way, it opens ion channels for potassium-rich endolymph to flow through. This eventually causes a depolarization. Then, when the hair cell jiggles the opposite way, it closes that ion channel, which eventually results in a hyperpolarization. So as we can see, these sound waves vibrate everything. The basilar membrane is really, really gelatinous. It's jello. And these hair cells are attached to the tectorial membrane, so we can see that the bottom is jiggling. So this oscillation of the transition between depolarization and hyperpolarization eventually results in a specific pattern of action potential messages to be fired along our cranial nerve 8, or our auditory nerve, or our vestibulocochlear nerve. So think of this process as a person that's playing the piano. So each of these sound waves activates a specific type and number of these receptors. When these receptors fire on and off, it's like a person playing piano keys. Our brain takes these different action potential notes and then it interprets them to form a cohesive sound. It's like our organ of corti are playing little individual notes for us, and then our brain turns them into something that we can interpret and recognize. But how do we hear different frequencies? Or in simple terms, how do we hear deep low sounds and how do we hear really high pitched sounds? This is a great question, and the simple answer is that some of our hair cells are more stiff and need higher frequencies to make them jiggle, while other hair cells are very, very loose and will jiggle even at low frequencies. So once our sound has been transduced, where does that action potential information go? Well, we know that it travels from our receptor cells, shown here in blue, to our afferent neurons, which make up the 8th cranial nerve, the auditory nerve, the vestibulocochlear nerve. Now, you're going to ask, why are all these science guys always making these really hard names that no one can remember? Well, just remember that this is the part of the inner ear that gives us our vestibular sense, and this is the part of the ear which we call the cochlea, so that is why it is called the vestibulocochlear nerve. So after we transduce in the cochlea, we know that the 8th cranial nerve eventually will end up in our auditory cortex. This is the part of the brain that interprets our sounds. So what have we learned today? We've learned that sound pressure, or how loud something is measured in decibels. And we know that the frequency of sound, or how high pitched a sound is measured in hertz. We learned that sound waves enter our ear canal and then vibrate our eardrum. This eardrum then vibrates our auditory ossicles. This eventually vibrates our cochlea. We know that inside of the cochlea, there are little itty bitty hair cells that have a jiggly base and a hard roof. We know that different sound frequencies and decibels cause the hair cells to jiggle. This allows cations into the receptor and will also block the receptor. This eventually causes action potential messages to be sent along our 8th cranial nerve, the auditory nerve, the vestibulocochlear nerve, which eventually ends up in our auditory cortex. So this is how our brain interprets sounds. Thank you all so much for taking the time to learn with us today, and be sure to like and subscribe for more content. Thank you.